All right, so I'd like to start by acknowledging the traditional owners and custodians of the land on which we're meeting today, the Wurundjeri people of the Kulin Nation, and pay my respects to the elders past, present, and the elders from other communities who may be here today, as well as the elders and traditional custodians of the lands where anyone who's tuning in remotely may be sitting right now. So it's my pleasure today to introduce James Fu. James received his bachelor's degree from Nanjing University, where he studied protein recognition with nanomaterials. And I remember the first interview that Tony and I had with James, it was clear that he had a really special combination of programming skills and domain knowledge and understanding of biochemistry that was going to really help him succeed as a PhD student. And we were lucky to have him join the lab to explore his growing interest in machine learning, being supervised by Tony, Justin Vito, and myself. And although James knew how to program, he was really new to bioinformatics. And he's done an excellent job of developing his skills and understanding in bioinformatics and related areas, while also adapting to the many challenges posed by the switch to remote work and doing a PhD during the pandemic. And he's been a really resilient and enthusiastic student who always has new results and figures to show us during supervision meetings, sometimes more figures and results than we can get through in the given time. And he's taken on a really ambitious project that combines biochemistry, structural biology, bioinformatics, statistics, machine learning, software engineering, clinical genetics, and genomics. And I think you'll see a little bit of all of these things today as James describes the challenges, opportunities, and insights that he's derived from working with data and high-throughput mutagenesis, and all of the diversity that exists in these various studies of a large amount of different interesting proteins. And so with that, James.
All right, thank you, Alan. Thank you for the lovely introduction. And thank you everyone for coming to my completion seminar. So today I'm going to talk about understanding the protein variants with high-throughput mutagenesis and machine learning. So my PhD project is about protein and protein variants. According to the central dogma that a protein coding gene can be transcribed and translated to a chain of amino acids, which will then folded and combined with other molecules to become a protein. Protein as the major functioning molecule in an organism, it may work as a transporter, carrying waste or nutrients. It may also work as an enzyme, catalyzing specific biochemistry reactions. The normal functionality of a protein sustains the survival of the host cells. However, the protein sequence is changeable. Missense variant is a type of genetic variation that can change the amino acid in a protein sequence. And many of these variants can be functionally neutral. However, some of them may potentially change the protein function greatly. For example, some of these variants may cause the target protein fold incorrectly, and some may happen on the enzyme active sites, decreasing the activity of the enzyme. And some may eventually cause the death of the host cells. Although these missense variants can potentially change protein function greatly, the impact of most of them is still unknown. With the rapid development of whole genome sequencing, it is now much easier to identify variants in human genome. For example, according to EXAC, which is an online database for human genetic variation, there are more than 7 million human variants identified in human genome so far, and 3 million of them are missense variants. However, only 1/10 of these missense variants has been interpreted in ClinVar, which aims to interpret the clinical importance of these variants. And to make things worse, over 70% of the variants are variants of uncertain significance, which means even though we have identified these variants in the genome and try to analyze them, we still have no idea if they are going to cause the disease or not. So, evaluating the impact of these missense variants is becoming more and more important. And to do this, researchers have been using mutagenesis experiments to evaluate these variant effects for many years. Most of these experiments are low in throughput, which makes it possible to measure specific protein property individually for each variant. For example, If a variant is causing the change of cell growth rate, this can be measured by counting the number of a cell within each colony on a culture dish. And for variants that change enzyme activity, this can be measured by quantifying the enzyme reaction product by approaches like Western blot. Among all these mutagenesis approaches, alanine scanning is one of the mostly widely used approach. And in such experiment, the researchers will try to substitute all the wild-type residues to alanine within the target protein region, and then measure the effect of all their variants by some appropriate assays. The reason for choosing alanine is that its structure can be regarded as the backbone for most other amino acids, and by substituting to alanine, the researchers can evaluate the importance of the wild-type side chain. And in recent years, another type of mutagenesis experiment has been developed, which is named as DMS, short for deep mutational scanning. In a DMS experiment, the researchers can evaluate a huge number of variants in just one single experiment. Compared to alanine scanning, which involves the systematic substitution to alanine, and then with the variant effects measured in a low-throughput assay, in a DMS experiment, instead of just alanine substitutions, the researchers will try to create all possible single amino acid substitution in the target protein region, and then measure the effect of all these variants in a single experiment, usually by high-throughput sequencing. So here is a general workflow for a DMS experiment. In this setup, the target protein or target gene is essential for the survival of the host cells. And the first thing to do is to create a full DNA library containing all possible single amino acid substitution in the protein product. Then this library will be introduced to the cell line with the variant protein being produced. And the cells will be cultured for a certain amount of time, and which is known as the selection process. And in this process, cells with functioning variant will be enriched, and cells with non-functioning variant will be depleted. The variant frequency before and after selection will be determined by high-throughput sequencing, and the ratio of them will be calculated as DMS scores, which indicates the general fitness of each variant. For example, in this case, higher DMS score means the variant is enriched in the process, and so they are functioning. Otherwise, if the DMS scores are lower, it means the variant is depleted in the process, and so they are causing loss of protein function. The DMS results are usually visualized to a heat map looks like this. So in this case, each row represents a protein residue or a position, and each column represents a variant amino acid. Each pixel in this figure represents a single missense variant. For example, for the pixel on the upper left, it represents the alanine on residue 150. It's substituted by tryptophan. The color of the map shows the DMS scores, and in this case, white means the variant is behaving just like wild type, and blue means the variant is causing loss of protein function, and red means it is related to gain of function. Instead of looking at each variant or each pixel individually, this result can also be read horizontally or vertically. For example, if you read it horizontally, you can find certain residues that are sensitive to almost all substitutions. And if you read it vertically, you can find certain variant type amino acid, for example, proline in this case, that will also always cause loss of protein function, probably due to its unique shape. Well, in certain cases, there will also be missing values within a DMS dataset, as indicated in these gray boxes in our example. The reason for this is mixed, that sometimes this variant may be missing from the initial library, and sometimes they may just didn't pass some quality control. And the first aim of my PhD project is exactly about dealing with these missing values, that I want to develop and benchmark imputation tools to impute the missing values in a given DMS dataset. And to do this, we started with collecting some DMS data so we can have a better understanding about how many values are missing within each dataset and use such data to build our models. We collected the DMS results from MapDB, which is developed and maintained by Alan. And we went through all the experiments within it to find suitable data to be included in our analysis. And then we normalized all the DMS scores for each dataset to make sure that they are comparable to each other. Our final DMS dataset contains results targeting 61 unique proteins measured by 146 DMS experiments and covering over 400,000 B-sense variants. So now we have our full DMS datasets available. It's time to have a look at how many values are actually missing within them. To do this, we calculated the completeness for each DMS dataset by the ratio of number of measured variants versus the number of expected variants. And the latter one is calculated as the number of scanned residues times 19, because there are 20 amino acids in total, minus 1 wild-type amino acid. The result is shown in this histogram that from the rightmost bar, you can see around 30 DMS studies in our dataset is fully completed. However, most DMS studies have a completeness between 90 to 100%, and many of them are having even lower completeness. And many of them are targeting the human proteins. So this result kind of indicates the necessity of a proper imputation tool for DMS results. So far, according to our knowledge, there are at least 2 methods developed for imputing the DMS scores. And these methods use certain protein features as input to model the incomplete DMS dataset and then impute the missing values within it. To be more specific, these protein features are used like evidence. They basically shows if the variant has been observed in the protein homologs or where the variant is located in the protein structure. And then this evidence will be used to explain or fit the DMS scores measured experimentally with a certain machine learning algorithm. And in the imputation process, these protein features can be applied to the model again to impute the missing values. However, one issue about this method is the availability of these protein features. For example, in this case, the features are only available for 2/3 or half of our DMS datasets and for these 2 methods. So we wondered if we can build a protein feature-free imputation method by just learning the DMS scores themselves and takes no extra protein features as input. So such a method can be applied to all the DMS datasets we have. And to do this, we must rely on the connection between DMS scores. So like I mentioned, a DMS result can be read vertically, and I also want to mention that there are some shared information between columns as well. For example, arginine and lysine are both positively charged amino acids, and the substitutions to these 2 amino acids may usually have similar DMS scores. So we can somewhat confidently impute the missing values in the lysine column by the values in the arginine column. Extending this idea, we built our first imputation method named AA-lasso. In such a method, we pick 1 variant type amino acid at a time, which is the AA comes from in the name of the method. And then we train a lasso regression model to impute the missing value in the picked AA using DMS scores from the other columns. Then we iterate over each AA type until all the missing values have been imputed in such way. So this AA-lasso method impute missing values based on the connection between each variant types, or in another word, between the connection between each columns. But we can also expect that there are connections between rows as well. So to simultaneously capture the connection between columns and rows in a DMS dataset, we build another imputation method named factorized DMS. In such method, the sparse DMS score matrix will be approximated or factorized by 2 lower rank matrices, and the multiplication result will give us the imputed full score matrix. The underlying biological intention of this method is that we believe these 2 lower ranked matrices can capture the connection between each protein residue and each variant amino acid individually. So now we have built 2 imputation methods already, and it's time to evaluate their performance and benchmark them with published approaches. To do this, for each DMS dataset, we first mask 10% of data later used for testing, which can be regarded as the fake missing values. And the rest of the data were used for training the imputation methods. Here, we are comparing 5 imputation methods. The 2 were developed by ourselves, 2 previously published, and 1 is to impute the missing values with mean DMS score on the same protein residue, which is frequently used in a DMS analysis. After the models were built, the variant scores in the masked testing data were imputed by this method and compared with the experimental results by calculating their Pearson's correlation. We repeated this process for 5 times and iterated over each DMS dataset. And here in this figure, I'm showing the benchmarking results. On the horizontal axis, we have different imputation approach. And on the vertical axis, we have the imputation performance measured by Pearson's correlation. Each dot in the figure represents a tested DMS dataset, and the black line in the middle shows the median performance of all these datasets. A first observation is that for these published predictors or published imputation methods, we observe much less dots within them because not all our DMS datasets can be applied to these methods due to the feature availability issue. And on the other hand, for our method, we have all the 146 DMS results available. And when looking at the median performance, you can see that the residue mean approach gives the lowest imputation accuracy. However, the published Envision method as well as our A-lasso method gives the highest overall imputation accuracy. To further analyze the advantage and disadvantage between these 2 best results, we further look at the details of them. Here we calculated the correlation improvement by using the Envision method against the A-lasso results as shown on the vertical axis. And positive value means the Envision method performs better, and negative value means the A-lasso method performs better. In this case, each dot still represents a DMS dataset, and we color them by the completeness of the data. So you can see that when the datasets are having generally higher completeness, then the 2 methods tend to perform similarly with most dots around 0. But for datasets with lower completeness, there's a trend that the Envision methods tend to perform better, although with 2 outliers down here, which is probably because they are lower size of data as we checked, and this result can be just noisy. Anyways, to systematically understand how this data completeness can affect our model performance, we try to repeat previous benchmarking analysis in a slightly different way. In this case, we're only including highly completed DMS data for analysis, and still 10% of data were masked for later testing, but in this case, a gradient of the rest data will be fed for training, ranging from 10 to 90% of the full DMS dataset. And the results shown here on the vertical axis, we have the imputation performance, and on the horizontal axis, we have the proportion of data used for training, which is essentially the same as the data completeness measurement. And as you can see, our A-lasso method and the factorized DMS gives the highest imputation performance, while more than 80% of data were used for training. However, their performance dramatically decreases when the data completeness is decreasing. Well, this makes sense, because if the data are highly sparse, then there's no score connection to be learned from these 2 methods. Well, on the other hand, for these published protein feature dependent approaches, they can perform reasonably good when only 10% of the data were used for training. And this indicates that they are more suitable for highly sparse DMS datasets. So we can kind of expect that each imputation method may have their own strength or advantage. So I wondered if we can build an ensemble method that can aggregate all these 5 imputation results to a single one, which can potentially outperform each of them. The possible easiest way to do this is just to take the mean or median of these 5 results to become our ensemble result. But here what I did was actually slightly more complicated than that. That I trained a random forest regressor, which takes the imputed scores and high important protein features as input, and it was trained on cross-validated training data to avoid overfitting or data circularity issue. Well, if you are interested in the details, so I'm happy to take any question after the talk. But if this ensemble method confuses you, please just assume that it takes the mean or median of these 5 results to become the final ensemble result. And then we decide to have a look at the performance of this ensemble method. So here we are comparing the ensemble method with individual imputation approach by calculating the correlation improvement by using the ensemble one against the individual ones. And here, positive value means the ensemble method performs better, and negative value means the individual approaches are better. The middle line for the boxes indicates the median improvement for the tested DMS datasets. And as you can see, our ensemble method can improve each individual approach in terms of the median improvement because they are all above 0. And to further torture myself, for each DMS dataset, I picked one individual result that gives the highest imputation performance as the individual best result, as shown in this last purple box, and compared it with the ensemble ones. So in this case, the ensemble result still shows similar, if not better performance compared to the individual best result. So here we conclude that our ensemble method can improve the overall imputation accuracy compared to all these individual approaches. And that's all about the first story